Okay, so we're now in chapter 4, extensions of Mendelian genetics. And so in this chapter, we're going to take all the principles that we learned in chapter 3 and apply them to situations where things are a little more complex. So in chapter 3, we learned about classic Mendelian ratios, and we learned that, if you take two heterozygotes and cross them, exhibiting one trait, you'll get a 3-to-1 ratio between the two traits and a 9-to-3-to-1 ratio for dihybrids. However, these ratios will be modified if some more complicated things are occurring. So, for example, if these genes aren't straightforward, dominant, and recessive, that can change the ratios. If you happen to have more than one pair of genes influencing a particular character, so for example, in humans, our eye color, there are, I think, something like 50 different genes that can affect eye color. So that makes it a lot more complicated. And then if you have more than 2 alleles exist in a population for one particular trait, that can also change the ratios. And so that's the kind of things we're going to be discussing. But even though these ratios are modified, Mendel's basic principles of segregation and independent assortment will still hold true, and you'll see that in the examples that we're going to go over. Now, the things that we're going to look at, the first thing we're going to talk about is incomplete dominance and codominance. We're going to talk about multiple alleles, how genes interact with one another and how that affects the ratios. We're going to talk a little bit about genes that are present on the X chromosome, and then how the environment also influences gene expression and phenotype. So once Mendel's principles were kind of rediscovered, then researchers started studying, the way that genes influence phenotype, and these investigations were called neo-Mendelian genetics. And these researchers determined that there's a lot of situations in which the simple inheritance patterns that Mendel described are not followed. So before we start talking about that, I want to talk a little bit about just what it means to express a gene. So we know a gene is a segment of DNA that codes for the production of a particular gene product, and that gene product is usually a protein, or is always a protein. So the gene gets translated into RNA, gets transcribed into RNA, and then that RNA gets translated into a protein product. And usually those proteins are enzymes. And then we know that enzymes are proteins that carry out chemical reactions in the cell. Now, when we say that a gene has been expressed, what we mean is that an RNA copy has been made of that gene, and then that RNA copy has been translated into a protein. So we have produced the gene product of that gene. Now, every cell in our body pretty much has the same genetic information. So your skin cells, your muscle cells, your liver cells, your pancreatic cells, all cells have the gene for insulin, for example. However, it's only the beta cells on your pancreas that actually makes insulin. So it differs which genes are turned on and which genes are turned off in various cells, and sometimes at what point those genes are turned on and off, and then if they are turned on, how much product is made from that gene. All these things can vary. The expression of genes is a very highly regulated, complex process. So that certainly can affect phenotype based on when genes are being expressed and how much gene product is being produced. And, like I said, that's going to influence the phenotype significantly. Okay, so let's talk a little bit about alleles. So we know that alleles are just alternate forms of genes. We kind of typically think of a normal allele in a population as being the wild type allele. This is the one that's most frequent. Usually this is the dominant, but not always the dominant allele. And so we consider this the wild type. Mutant alleles are going to be variations from that wild type allele. They're going to be altered DNA sequences, and usually If they affect the phenotype, if the mutations affect the phenotype, you either have a loss of function or a gain of function. So a mutation is just simply a change in the DNA sequence. So if you change the DNA sequence, you can potentially change the sequence of amino acids in a protein. And that can affect the way that protein folds, and its function is tied very tightly to its shape. So if it's not folded properly, it can actually cause the protein to completely lose its function. In that case, it's called a null allele, or maybe just reduce its function, make it where it's not quite as functional as it should be. Like maybe it doesn't bind to its substrate as well, so it doesn't carry out the chemical reactions as quickly as it should. Usually these loss of function mutations create recessive alleles. And so a good example is cystic fibrosis. This is due to mutations in a protein that involves the transport of chloride ions out of the cells. And you have this high incidence of chloride ions inside of a cell, which is really the whole problem and causes issues with water. It brings water into the cells, removes water from the mucus in the respiratory tract, and the mucus gets really thick. And so that's one of the main symptoms of cystic fibrosis, and it's just due to this protein that's not folded properly. And that's a recessive mutation. If you can also have a mutation in the regulatory sequences of a gene, which is usually right before the coding sequence. And this is where the enzyme that's going to carry out transcription binds to the DNA. And if it doesn't bind and doesn't transcribe the gene, or if it binds too readily and creates too many transcripts, this can lead to something called a gain of function or enhanced function mutation, or sometimes maybe it's a different function. But anyway, it changes the gene expression pattern. The protein that's made is the same as if, you know, the protein itself is not mutated. It folds the way it's supposed to and everything. But it's just being expressed when it's not supposed to, or it's being expressed more than it's supposed to. And so these are usually dominant alleles, meaning you only have to have one in order to exhibit the phenotype. So classic dominant recessive gene expression, this is what we went over in chapter 3. So this is what Mendel saw. He saw that if you could either be homozygous dominant for a trait, meaning you have two alleles for the dominant trait, like for example tall in plant height. You can be heterozygous. If you're heterozygous and you have one dominant and one recessive allele, it's going to look phenotypically the same as a homozygous dominant, so it's also going to be tall. And then if you have homozygous recessive, meaning you have two of the mutant alleles, then you're going to have a different phenotype, and so you'll have the dwarf plant. So you only have the dwarf plant if you have a homozygous recessive situation. Now, a molecular explanation of this is simply that there's this growth hormone that's involved in producing height of the plant to a certain level. As long as you have one allele of that hormone, you're going to reach a certain height with the plant. If you don't produce that hormone, then the plant's not going to grow like it's supposed to, and so it will be short, or a dwarf plant. So the recessive short or dwarf phenotype is due to a loss of function because you don't produce this growth hormone that's necessary to make tall plants. Now, typically, we don't see that a single trait is influenced by a single gene. Usually there's multiple gene products involved. For example, let's say you're going to make an amino acid. You're going to synthesize an amino acid. It might take 10 different enzymatic reactions, all involving 10 different proteins, or 10 different enzymes, 10 different gene products to carry out that synthesis pathway. If any one of those enzymes is mutated and doesn't function properly, you're not going to produce that end product. You're not going to produce that amino acid. So the phenotype might be that you don't make this particular amino acid, and maybe in the case of a bacteria, that might mean that that bacteria dies because it can't make an important amino acid. But it could be different mutations occurring on different alleles of different genes that cause that same end effect. Because the mutations in any of these enzymes gives you that same end problem, which is you don't produce the end product. A good example is eye color and Drosophila, and we're going to be coming back to this a lot in this chapter because a lot of the things that we talk about are what Thomas Morgan studied, and he studied these things in fruit flies. So eye color is one of the first things that Thomas Morgan recognized, and we're going to talk more about eye color when we get to X-linked traits. But the eye color of a fruit fly is actually from two different pigments being produced and then deposited into the eye. And if anything happens with these eyes, so the wild type color is brick red because of the mixture of the brown and the bright red pigments. But if anything happens through any processes of making these pigments, either one of them, it's going to lead to different eye colors. And specifically in the brown pigment, if you don't make brown pigment, the eyes are going to be bright red. But there might be 5 different enzymes involved in the production of that brown pigment. And if there's a mutation in any one of those 5 enzymes, you're not going to make the brown pigment. Alright, so when Thomas Morgan did his studies, he decided to deviate a little bit from the way Mendel wrote the symbols for alleles. So Mendel, we've already talked about how he wrote things. He had the capital letter for dominant, the lowercase letter for recessive, and usually the letter came from the recessive trait. So for example, with tall and dwarf plants, you would use the letter D for dwarf, capital D was for tall, and a lowercase d was for dwarf. When Thomas Morgan started researching the fruit fly, he was studying mutants. So if a fruit fly had short wings, or had a different color body, or had the wrong color eyes, anything other than what we would consider the wild type, those were referred to as mutants. And so he distinguished between mutant versus wild type. And so for example, body color, an E was written for ebony color, where the wild type color is gray. So if you had a lowercase e with no plus sign, then that's ebony. But if you had a plus sign with the lowercase e, then that was wild type, which would be by default gray. And you would use lowercase letters if the mutant was recessive, or uppercase letters if it was dominant. You'll get used to this. You'll see we'll use a whole lot of different symbols throughout this chapter and the next chapter on through, probably through chapter 7, and then when we move into molecular genetics, we'll move away from this. But you'll get used to all the different symbols. Now, in some cases, there are 2 alleles where there is no dominant or recessive condition, and we'll talk about those in a few minutes. So sometimes you'll have 2 uppercase letters and then some kind of superscript to differentiate between the 2 alleles. Sometimes you have situations where you have dominant alleles and recessive alleles, and some of the dominant alleles, we have multiple dominant alleles. So in that case, you might do a mixture of both, where you have the uppercase letters for the dominant and lowercase letters for the recessive, and then the superscripts to differentiate between the dominant alleles. All right, so let's start talking about some of these situations. One of the situations is incomplete dominance. This is where you have, it's also called partial dominance. This is where you have, when you take 2 alleles, like what you would consider normally, if normal inheritance, Mendelian inheritance, you have like a dominant and a recessive allele. When you put the 2 together and create a heterozygote, you get, it looks just like the dominant one. But in this case, when you take 2 alleles and put them together, you get an intermediate or blended phenotype. So it's in between the two. So for the example we have here is you have red flowers. One type of allele produces red flowers. The other allele produces white flowers. And if you produce a heterozygote, you get pink flowers. So you get something in between red and white. So in that situation, when you cross two heterozygotes, instead of getting that 3-to-1 ratio, in this case, let's say if red was dominant over white, you would expect to see 3 red flowers and 1 white flower. 75% of them red and 25% of them white. Instead, we see that you have 25% red, 25% white, and 50% pink. So all the heterozygotes have their own separate phenotype from the homozygous ones, which is also identical to the genotypic ratio. Now the way this works is the allele for white flowers is probably some kind of loss of function mutant. So it's probably that you have this red pigment being produced, which is what makes the flowers red. A white mutant doesn't make that pigment, so by default it's a white flower. And if you only produce half the pigment that you normally would produce because you only have one allele for the pigment, then instead of it being red, it's going to be something lighter than that, which would be pink. So this means that the effects of this gene expression is actually quantitative. So it takes a certain amount of this pigment to create red flowers. And when you only have half that pigment being made because you only have one allele being expressed, then you end up with the pink flowers. And the amount that it would take to get to the red flowers is called the threshold. And we see this even though we don't actually see it. It still exists even in complete dominant recessive traits. An example is in Tay-Sachs disease. Remember we talked about that last chapter where you have the hex A enzyme that breaks down the lipids in the neurons, and if those lipids aren't being broken down, they reach toxic levels and actually kill the neuron cells. As long as you have one allele that codes for the enzyme, you don't have the disease, and it's only these homozygous recessive individuals that pick up 2 mutant alleles that end up with the disease. But we do find that actually these heterozygotes only produce half of the enzyme. Whoops, I went too far. But anyway, these heterozygotes only produce half of the enzyme. They just don't, it's just enough to prevent the disease because half the enzyme meets that threshold. Okay, so now in codominance, this is different from incomplete dominance. With codominance, both alleles are clearly evident in a heterozygote. So they're expressed equally. So this isn't an intermediate phenotype. This is a new phenotype in which the 2 alleles are seen equally. An example is the MN blood group. Now this is different from, if you know, like when you give blood and they ask you your blood type and you say you're type O. This is a different set of information about blood groups. But the MN blood group, you have there's 2 alleles in the population. There's M and N. If you can either be homozygous dominant for M, or not dominant, excuse me, homozygous for M, homozygous for N, or you can be a heterozygote where you have both M and N. So this is a codominance where the 2 are seen equally. So if we go back to our incomplete dominance example with the pink flowers, with codominance you see both colors equally apparent. So like in this flower you can see there's white and red, but it's not pink. It's just some portions are red and some portions are white. Both of the colors are equally apparent. With the incomplete dominance, you have a blend of the 2 colors to make a pink flower. So that's the big difference between codominance versus incomplete dominance.
In his studies involving pea plants, Gregor Mendel described phenotypic traits in which one allele was dominant to a recessive allele. He showed that with complete dominance, the phenotype of the heterozygote is the same as that of the homozygous dominant. Since Mendel's discovery, we have seen that many allelic pairs do not show complete dominance. When one allele is not completely dominant to another, it is said to show incomplete dominance. The heterozygote is intermediate in phenotype to either the homozygous dominant or homozygous recessive condition. One example of incomplete dominance occurs in the color dilution gene in horses. The horse color phenotypes chestnut, palomino, and cremello are controlled by the color dilution gene. Chestnut, also called sorrel, has a red pigment over the body and in the mane and tail. Cremello is extremely light-colored, almost white. Palomino appears as an intermediate between chestnut and cremello and has a golden body color with a flaxen mane and tail. The explanation for these different coat colors involves two alleles, big C and big C-CR. A chestnut horse is homozygous for the big C allele. A cremello horse is homozygous for the big C-CR allele. A cross between a chestnut and cremello produces all heterozygous offspring, which are palomino in color, big C-CR. The gene symbols used here do not imply dominance or recessiveness. Neither allele is dominant or recessive to the other. Palominos are not true breeding. A cross between two palominos always produces chestnuts, palominos, and cremellos in a 1 to 2 to 1 ratio. Notice that the genotypic and phenotypic ratios are identical. This 1 to 2 to 1 phenotypic ratio is characteristic of a cross between two heterozygous individuals. Another modification to Mendel's complete dominance is codominance. This occurs when the heterozygous individual exhibits the phenotype of both homozygotes. This differs from incomplete dominance, in which the heterozygous individual exhibits a phenotype intermediate between the two homozygotes. In codominance, there is no dominance or recessiveness of an allele. Instead, two dominant alleles are fully expressed at the same time. An example is the human MN blood group system. The MN alleles result in the formation of antigens on the red blood cell surface. There are two alleles in the MN system, LM and LN. These alleles combine to form three blood types, M, MN, and N. These blood types are determined by the genotypes LM, 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 LN, and LN, LN, respectively. A person homozygous for the LM allele and another person homozygous for the LN allele can only produce offspring who are heterozygous, LM, LN. These offspring will show both the M and N antigens on their red blood cell surface. Two heterozygotes produce offspring who have the M, MN, or N phenotype in a 1 to 2 to 1 ratio. The genetic segregation patterns of codominant and incompletely dominant traits are the same, 1 to 2 to 1 phenotypic and genotypic ratios for progeny from crosses between two heterozygotes. However, heterozygotes with two incompletely dominant alleles have a phenotype that is intermediate between the phenotypes of the two homozygotes. In contrast, alleles that are codominant are each fully expressed in heterozygotes, so that heterozygotes show the phenotypes of both homozygotes.
All right, multiple alleles. This is a situation in which in the population there's more than just two alleles present. So everything that Mendel studied, the seeds were either wrinkled or round. They were either yellow or green. The plants were either tall or short. There was always two alleles and two phenotypes for each trait that he studied. But in more complex situations, like in humans, there are going to be a lot of situations where there's several alleles present for one particular gene. But just keep in mind, there may be 50 different possible alleles, but each individual is only going to have 2, because you can only inherit 2 alleles, one from each parent. Now, this is actually the proteins that are found on the surface of your cells when they do tissue typing. So if you need a bone marrow transplant or a kidney transplant or something like that, and you go on the UNIS waiting list, they have to find a match for you. And that's because there's so many different alleles for these different proteins in the population that it's hard to find that one person that has the exact same proteins as you. So these genes that have a bunch of different alleles are often referred to as polymorphic. So how do these multiple alleles arise? Well, what we consider the wild type allele is the one that's just most common in the population. And then mutations occur over time that lead to this allelic variation, which is really what gives us this genetic variability, which is part of why we all look so different. The ABO blood types is a really good example of multiple alleles. This guy, Karl Landensteiner, received the Nobel Prize for Medicine in 1930 for his findings studying the blood types. He noticed that some people had severe reactions after a blood transfusion, called a transfusion reaction. He wanted to figure out what was causing that, and so he was able to discover the ABO blood group system through his studies. Okay, so we know that there are 3 different alleles that influence blood types. You can have the type A allele. This allele, so there's this substance on your cells called the H substance, and that's just kind of like the base substance to which these carbohydrate extensions are added. Type A is one particular type of carbohydrate that gets added, and type B is a different type of carbohydrate that gets added. So if you have the A allele, you add the type A carbohydrate. If you have the B allele, you add the type B carbohydrate. And if you have the lowercase I allele, then you don't add any carbohydrates, and you're left with just that kind of base H substance on your red blood cells. Now, the lowercase I allele is recessive to the A and B alleles, but the A and B alleles are actually codominant to one another. So that produces 4 different blood types. You can either be type A, meaning that you have, you do produce the type A carbohydrate extension on your blood cells. You can be type B, meaning you produce the type B carbohydrate. You can be type AB, meaning that you have both types of carbohydrates. Or you can be type O. With type O, all you have is the H substance. You don't add either carbohydrate to your cells. Okay, so let's say you go to the hospital and you have to get a blood transfusion. It's really important that you get the right type of blood cells, because these different carbohydrates that are present on the surface of these blood cells are actually antigens. And antigens are substances that cause the body to produce antibodies. These antibodies are produced against foreign antigens, not self-antigens. So if you have type A blood, then you won't produce antibodies to the type A carbohydrate, because your body sees that as a self molecule, a self-antigen. However, if you have type A blood and you get type B blood transfused into your body will see that type B carbohydrate as being foreign and will produce antibodies against it. Now, when that happens, the antibodies actually bind to the red blood cells and then kind of corral them all together, causing them to agglutinate, and which creates blood clots, which are never a good thing to have. So, and we can actually use this agglutination reaction to determine a person's blood type. So we don't have to take their DNA and sequence the DNA and figure out what alleles they have. All we have to do is take their blood and do these agglutination tests to see which types of antigens are present on the surface of their cells. Type AB people are considered universal recipients because their bodies see both the antigens as self. So they don't produce any antibodies against any of the blood types. So someone with type AB blood can receive type A blood because they already have the type A antigens, or they can receive type B blood, or they can receive type O blood because the O blood doesn't have any of the antigens. All it has is that base H substance, so that, there's no problems there. And because of that, type O people are considered universal donors because there's no antigens in the blood to cause a reaction. The white locust in Drosophila is another example of multiple alleles. Drosophila are supposed to have that brick red eye color. If they have two recessive alleles for white eyes, they won't transfer any pigment to the eyes, and so the eyes will be white by default. There's actually 100 different alleles that all create the same problem, which is these are different mutations that prevent the pigment from being carried to the eyes. Like I said, this is a transport situation. So this is a protein that actually transports the pigment to the eyes. There's other mutations in the actual creation of the pigments that can lead to white eyes as well. If the white eyes aren't, if the pigment's not made, then again the eyes could potentially be white. All right, the last type of alleles that we're going to talk about are essential genes and lethal alleles. So essential genes are, as the name suggests, genes that are essential for survival. So, and as long as you have one copy, usually you're fine. An example is the hex A gene that codes for the gene that codes for the hex A enzyme that the Tay-Sachs, people with Tay-Sachs are missing. So if you're missing those alleles, if you're missing both of the alleles for an essential gene, then it can lead to death. And so these are considered lethal alleles for that reason. However, there are lethal genes that can also be dominant, where only one copy of the allele is necessary to cause death. Huntington's disease is a good example of that. And we'll talk more about Huntington's disease in later conversations. All right, so a good example of lethal alleles is the one associated with yellow coat color in mice. So mice are typically this agouti color that you see here. It's kind of like a blend of yellow and brown, and it comes from this banding pattern that you see here, where a band of yellow pigment is laid down on the shaft of the hair. So the hair is black, and then it has this yellow band deposited on the black hair, and it creates this agouti color. And so mice that are agouti are actually homozygous for that trait, for that laying down that yellow pigment. It is a mutant allele called the AY allele. These mice, if a mouse carries only one of these mutant alleles, they're actually yellow all over. And so here's a picture of a mouse that you can see that's yellow all over. And he's fat too, but I don't think that has anything to do with the coat color. But anyway, this allele is dominant, so you only have to have one of these alleles in order to be yellow. And it turns out all yellow mice are heterozygous. They never breed true. You never find a mouse that is homozygous for the AY allele because these mice do not survive. They actually die during fetal development. So in this case, this allele behaves as a recessive lethal allele. All right, so see here, if you cross two agouti mice, you're always going to produce nothing but agoutis. So agouti mice always breed true, and they're perfectly healthy mice. Yellow mice, however, if you take two yellow mice and you breed them, they're always going to come out agouti and yellow. They never breed true, so they're always heterozygous. And note that normally, if you took two heterozygotes and crossed them, you would have that 3-to-1 ratio. But instead, we see a 2-to-1 ratio of yellow to a Agouti. And that's part because this lethal allele here, these mice die. They don't survive. So that's where we get that 2-to-1 ratio instead of the typical 3-to-1 ratio we would expect. And if you cross an agouti mouse with a yellow mouse, it's pretty much like a test cross because you're taking a homozygous for agouti, which in this case is recessive to the dominant Y allele for coat color, and so you get a 1-to-1 ratio of agouti to yellow. So here you can see for coat color, the Y allele acts dominantly, but for lethality, it's a recessive allele. So just some explanations of how this works. This is actually a deletion mutation, and the deletion removes part of this regulatory portion of the gene that codes for the enzyme associated with this yellow pigment. So when the regulatory part is removed, the gene is turned on all the time. So instead of just putting a yellow band on the hair, the whole hair gets painted yellow, and so these mice appear yellow. But the deletion, so the deletion includes this regulatory portion, but it also includes a big portion of the coding region of another gene that's close by. And that gene happens to be an essential gene, so that's why you never have mice with 2 alleles, because they die during development, and they die before birth. Right, the last thing, I think this is the last slide for this talk, that we're going to talk about is Huntington's disease. So this is a dominant lethal allele. The allele is called allele H. It's an autosomal allele. You only have to have 1 copy to result in death. And the most interesting thing about Huntington's disease is the onset of the disease is delayed, and usually people don't start showing symptoms until late in adulthood. Maybe sometimes as early as in their 20s, but usually people are in their mid-40s to early 50s before they even start showing any symptoms. And basically what happens is there's a breakdown of the brain matter, which you can see here, the big holes in the brain, which is going to lead to gradual nervous and motor deterioration. And offspring, because if you're a carrier for the allele, or not just carrier, if you have the allele, if you have the disease, then you have a 50-50 shot of transferring that allele to your children. So they have a 50% chance of inheriting the disease. Now the only way this works is if the people that have the disease can get to the age of reproduction to pass that trait down. So dominant lethal alleles are really rare. But Huntington's disease is one of those that the onset of the disease is so late in life that the trait does get passed down generations. All right, so that's it for this lecture. So answer the review questions, and then we'll move on to talking about how genes interact with one another.